Hello people, this is Kerry Anderson. Hope you all doing well. In today's video we will be talking about effect of insulin on human skeletal muscle protein synthesis is modulated by insulin. So if you like the video related to my health, I would like to request you all please do subscribe the channel, hit the bell icon or give thumbs up so that I will come up with more and more videos with lots and lots of information. So here we begin. Let's go. Effect of insulin on human skeletal muscle protein synthesis is modulated by insulin-induced changes in muscle blood flow and amino acid availability. Insulin is a potent anabolic stimulus for muscle protein. Insulin deficiency leads to a protein catabolic state with loss of muscle mass that can only be reversed with insulin therapy. Nonetheless, the mechanism by which insulin enhances muscle protein anabolism are still debated. A stimulatory effect of insulin on protein synthesis has been demonstrated in various tissues, including skeletal muscle. Furthermore, in vitro animal studies and a recent human experiment have shown that insulin can acutely stimulate muscle protein synthesis by increasing the initiation of mRNA translation. On the other hand, if the psychological increase in insulin secretion is pharmacologically suppressed during feeding in rat, the stimulation of translation initiation is abolished and muscle protein synthesis suppressed. Insulin can also reduce protein breakdown by stabilizing lysosomes and reducing the activity of the ubiquitin proteasome pathway. In human subjects, insulin infusion induces net amino acid uptake across the limb, forearm or leg, an indication of net muscle protein anabolism, but the mechanisms are still unclear. About half of these studies report that this effect was due to an increase in protein synthesis with no major changes or some reduction in proteolysis. Conversely, the other studies found a significant reduction in protein degradation with no significant changes in protein synthesis. Because these experiments were performed using comparable stable isotope artery venous balance methodologies, it is unlikely that technical and methodological problems were responsible for the conflicting findings. A review of these studies suggests that these apparent discrepancy on the metabolic mechanism by which insulin stimulates muscle protein anabolism might be explained by differences in amino acid availability for the muscle tissue. Specifically, all studies in which muscle protein synthesis had been stimulated by insulin also had an increased amino acid delivery to the muscle tissue. So, whereas most studies reporting a decrease in muscle protein breakdown with no increase in synthesis during insulin infusion also had a decrease or no change in amino acid delivery. The differences in amino acid delivery were mainly due to differences in amino acid concentration, which in turn were determined by the modality of insulin infusion and or the concomitant infusion of exogenous amino acid. This is because systemic insulin infusion decreases blood amino acid concentration unless amino acid are replaced by exogenous infusion in a leg or a forearm allows for the exposure of the muscle tissue to relatively high insulin level while avoiding a major reduction in blood amino acid concentration. The current study was conducted to determine whether the response of muscle protein synthesized to insulin depends on insulin-induced changes in amino acid delivery and availability for the muscle tissue. To test this hypothesis, we induced local hyperinsulinemia in one leg. In the absence of amino acid infusion, we used three psychological insulin doses, low, intermediate, and high, selected to either avoid a significant reduction in the systemic amino acid concentration, the two lower doses, or allow amino acid to decrease due to systemic hyperinsulinemia, because the reduction in amino acid concentration was expected to occur at the highest insulin dose. This design allowed us to determine the relative contribution of insulin dose and amino acid availability on the response of muscle protein synthesis to insulin. This design was preferred to a fixed systemic insulin infusion with and without amino acid replacement, because previous data indicate that available amino acid solution for intravenous infusion cannot exactly replace all essential amino acid and maintain the baseline physiological pattern. This is a major problem because it is known that essential amino acid can independently stimulate muscle protein synthesis. Thus, if the amino acid replacement infusion delivered excessive amount of certain essential amino acid, it would be impossible to distinguish between the effect of insulin and that of the amino acid infusion. In contrast, incomplete replacement of some amino acid should be responsible for an incomplete muscle protein synthesis response, again making data interpretation very difficult. Finally, the amino acid dose necessary to maintain the baseline concentration would be significant, so that over 3 it could reach 10 to 11 gram per subject, an amount that can stimulate muscle protein synthesis even in the absence of hyperinsulinemia. The protocol was designed to measure muscle protein and amino acid and glucose kinetic in the post-absorptive basal state 0 to 40 minutes and during insulin infusion the night before the study. Each subject was admitted to the General Clinical Research Center of the University of Southern California. At admission, a pregnancy test was repeated in women and a dual energy X-ray absorptiometry scan was performed to measure muscle mass. The subjects were then fed a standard dinner and a snack was given to 2200. After 2200, the subjects were allowed only water and Libitum until the end of the experiment. The morning of the study, polyethylene catheters were inserted into a forearm vein for tracer and dextrose infusion, in a contralateral hand or wrist vein for arterialized blood sampling, and in the femoral artery and vein of one leg for blood sampling. The arterial catheter was also used for the infusion of insulin and endocrine green. So that's all about the diabetes in this video. I hope you all liked it. So for signing off, before signing off, I would like to request you all please to like, share, subscribe as much as you can so that you will get the more knowledge about it. So that's all. We will definitely meet in the next video with my another content. Till then, God bless you. Bye bye and take care.